This is the last lecture in our high school workshop series for this semester. Today we're going to be covering the end of Foundations of Project Design, which we started last week. We're going to look a little bit at funding. We're also going to look at bioethics and a project that is closer to something that you all might be doing in the near future. For reference again, I'm Miranda. I am the project lead for the workshop series. And today I'll be teaching with Jenny, who is the former project lead and also the senior advisor to the SBI Club. Jenny, why don't you get us started?
Sounds great. Thank you, Miranda. So just a little recap of what we went over last week in the last lesson. We talked about the two different types, two very rough categories of the different types of scientific projects, the pure science projects and the applied science or engineering projects, and how the designs of these projects generally differ. And then we talked a bit about the where theory and reality can be different, in that something can work really well in theory, but due to the limitations of reality, it wouldn't actually work, and where it wouldn't actually work, or it would be difficult for it to work. And we took a look at examples where you can kind of predict that it might not work as well as theoretically it would ahead of time. And also we looked at an example where you really couldn't predict how well it was going to work until you actually engineered what you had wanted to create. And then we took a look at where exactly the pitfalls were in both of those cases. And we looked at how there was also parallel projects that had similar aims going on at the same time. So in this lecture, we're going to talk more about how exactly do you build a scientific proposal, because we've seen like a lot of the difficulties in how the planning works in the last lesson. And I will begin. Ooh. Next slide, yes. So I'm going to begin by talking about the basic components of a scientific proposal. So a scientific proposal is generally a pretty long document, and it has many different sections, but they can roughly be boiled down to 5 major sections: the background, the significance, the research methods, innovation, and references. So we're going to go through each of these components in a little bit of detail. You'll see I'll be referencing the research advisors at Columbia. At the bottom, I've had numerous really wonderful research advisors at my time at Columbia, and I've kind of synthesized all of the things that they've taught me and boiled it down and pulled out the most important points for this presentation. So I'll be referencing my most influential research advisors at the end of the section of the presentation, but just so you know like what that reference is for. So taking a look at the background section of a proposal, this is where you talk about the premise and the context of your project. So what exactly is the problem that you're trying to approach, whether this be developing a new therapeutic for cancer, or better understanding a cellular processes that might be important for tools such as gene editing. So what exactly is the problem you're trying to approach, and how bad is the problem? So that's a very informal way of saying how severe is this problem, and it kind of hints at the significance of the issue that you're trying to approach. So you really want to talk about existing work. What do we know so far, and what do we still need to find out or create? So with existing work, all scientific projects build on the previous projects that people have done. So what do we know about this field so far? What have others found? And why is it that this direction is still a good direction to investigate? Because what are the gaps in our knowledge? And then you also present the hypothesis. So now that we have a good idea of what we know so far, what people have done, what the gaps in the knowledge are, what do you think can be done in this direction? what do you think can be discovered or created? And this is largely based on existing work. So when I say based on existing work, I mean that you base your hypothesis on the work that has been done thus far. So based on existing data, based on data and results that have already been created, what do you think can be discovered anew? So for example, if a study, the first CRISPR study that, first mention of CRISPR was that there were these strange repeats in the bacterial genome. And then your hypothesis is maybe this is some kind of mechanism that protects the cell in some way. And I think we can discover what this mechanism does, and it's going to be useful. So this is really largely based on the work that has been done before, but it is also something novel, like something that we have not seen before, something that you want to discover or create anew. So for the significance part of the project proposal, it really addresses the question of, does the project address an important problem or critical barrier to the progress of the field? Is there a strong scientific premise for the project? And the strong scientific premise, it refers to both the impact, the effect of this project it would have if successful, and also on the previous data that will support your hypothesis is a good approach to take. It is a good direction to investigate in. And so this really assesses the effect of the project's success on the field. How would it improve scientific knowledge, technical capability, or clinical practice? So here you see it can approach either the expansion of our knowledge of how the world works with scientific knowledge, technical capability, creating tools that would help us develop new tools that we can use for various applications, and clinical practice. How can we use this in the real world? And the reason why clinical is in here, because this is referenced from the National Institute of Health's central resource for grant and funding information. But to make it more general, it can also be how exactly can this be applied in the real world? So what you really want to develop with your scientific proposal is a project that would impact a large or pressing field, in that it would have high significance. So I gave a few examples here: cellular metabolism and cancer. So cellular metabolism is something that, all cells go through. So understanding how this works would have a pretty large impact, in that if you discover a new way and a new mechanism that metabolism works, then potentially this can be used to understand other phenomena, other disease, etc. And this can also connect to cancer. A lot of the times when cancer occurs, it involves a misregulation of cellular metabolism. So cancer is a pressing field because there aren't too many really good treatments for cancer, and there are many different cancers that can't be treated too well, although there are some good treatments out there. So because of the enormous fatality rate of cancer and the number of people it affects, approaching cancer would give the project high significance. So for research methods, take a look at how you will achieve the aims of your project. So now that you have an idea of, this is what I'm trying to investigate, this is what I'm trying to discover or create, how are you going to do it? What cell lines or strains or animals or clinical volunteers will be involved? What experiments will you do, and what conclusions will you draw from them? So how will you set up your experiments, and what will you be able to conclude based on the data that you gather from those experiments? And what proposal reviewers will be looking for are the overall strategy, methodology, and analyses well reasoned and appropriate to accomplish the specific aims of the project, and how the investigators presented strategies to ensure a robust and unbiased approach as appropriate for the work proposed. So investigators here means the people leading the development of these projects, the scientists leading the development of these projects. So in this research methods, there's a bit of risk assessment. How risky is the project? And what I mean by how risky the project is how likely is it that what is proposed will actually be successful? So you could try to discover something, you could think that something is involved in this other processes, and maybe it's just not. Maybe there are some processes that are just not that influential. I mean, maybe you thought it was influential, but ended up not being influential. That's for the pure science side. For applied science, engineering side, it's that you're trying to create something. It'd be really awesome if you could create it, but in the end, you just can't get the components to work. Like you're trying all of these methods, but you just can't get the components to work. So how likely is it that your project will succeed? And this is where you start looking for failure points, because like to look at how likely it's going to succeed, you also look at how likely it's going to fail. So what reviewers look for is potential problems as alternative strategies and benchmarks for success for the project. And my advisors at Columbia have taught me that generally you only want one risky thing in the proposal. So you want, like, of all the things that you want to put in the proposal, all of these components need to work together in order for this to happen. You want to be sure that most of these components actually work. You actually have evidence that most of the stuff that you're relying on or the assumptions that you're making are true or do work. So in that case, you only want one thing that's kind of like, I'm not too sure if this is going to work, but if this works, it will be a really awesome thing. And that would be the thing that you try to like make it happen and optimize. And part of this risk assessment involves thinking about why people did not do it before you did. So one of my advisors had mentioned that whenever you come up with a scientific idea, you can pretty much assume that 10 people have already thought of your idea before you. So then the question is, why didn't they already do this? So there are a couple of possibilities, and I summarize them pretty generally here. First, someone actually tried to do what you wanted, you want to do, and it didn't work. So they never published it. Because negative results in science usually are not published. And by negative results, I mean results that show that something did not work. So because they're typically not published, if it doesn't work, typically you can't find it in existing scientific literature. And there have been movements to try to change that, where certain papers publish a lot of negative results, but generally speaking, negative results are not published. So the second possibility is that you lack resources. Like the person who thought of it just didn't have the money or the people or the equipment to do it at the time. And that like kind of is similar to the third point, but the third point focuses on lacking technology. And this is that the technology that is needed to make this project a success had not been developed yet. And that's why people didn't do it yet. And the last one is that new progress elucidated a new path for the research. Maybe it didn't seem like this approach or investigating this question will actually come up with something fruitful. It didn't seem like it was an interesting question. But now that there's been new progress and new discoveries have been made, it's more clear that you can actually find something more fruitful in this research. So now you start going on this path. And so what you really want to aim for is like projects that approach the last two. Lacking technology and new progress elucidated a new path for research. So if you lacked the technology previously, but now you have it, this is a really good time to do it. Because then now you have the resources to do what people like thought about doing before. And if new progress elucidated a new path, previously people didn't think it was interesting, but now we realize that this might be really fruitful. So this is now a good approach. If lacking resources is kind of like a circumstantial thing, in that like it really depends on the circumstances of the person. But like especially the first reason, if someone already tried it and it didn't work, you really don't want to do it again because it's still not going to work. So this is when talking to colleagues, talking to scientific advisors would really help. And talking to other people in the field and asking, like, have they considered this? Have they looked into this? Do you think this is a good idea? And maybe if they tried it before, like maybe they'll tell you, like, hey, I actually tried it before and it didn't work. So this is helpful here. All right, innovation. So the innovation, simply put, is a novelty of the project. So this is assessing whether or not the project has novel theoretical concepts, approaches or methodologies, instrumentation or interventions. And also, is it a refinement, improvement, or new application of theoretical concepts, approaches or methodologies, instrumentation or interventions? So with innovation, I've learned some really good tips from my advisors at Columbia. And generally, when you have a follow-up on a paper, so a paper had published like, oh, we found this, and you're like following up on it. For example, a group of people newly developed a therapy for some disease, and they developed it and they tested it on mice and it worked well. And you're saying, oh, let's test it on humans. So this is typically pretty low innovation. And usually you can kind of assume that the most obvious approach, the most obvious next steps for after the publication of a paper is already being taken by the group that published the paper. So when you have a follow-up on a paper, you kind of want to be quite careful in elevating your innovation to make it more novel. And two ways to make a project idea more innovative is that you take the paper that you were interested in and you connect it with another paper, like a new technology, a new finding, so that you can say like, oh, they're thinking about using this method to do it, but this new technology came out in another way to do it, and maybe we can use the knowledge from both to create a novel therapy that's different from either. Or another possibility is that you can focus on a surprising finding in the paper and investigate it further. And a good example of this is, in fact, CRISPR. When people first discovered CRISPR, it was just a really small footnote in one paper where it was like, we found these palindromic repeats. We found these repeats of these sequences in the genome. We don't really know what it is, and we don't know what it's about. So this is a pretty surprising finding, and you can investigate it further, and that would give it an elevated innovation. So you want to aim for high innovation projects. So I just wanted to give many thanks to my Columbia advisors for this section. And I've had the great fortune of meeting so many incredible research advisors. But the three people who've influenced me the most in terms of like developing as a budding researcher and also in terms of creating these slides are Dr. Harris Wang. So I work for Dr. Wang in his lab. I've been working for him for the past 3 years. He's absolutely wonderful. He is an assistant professor in the Department of Systems Biology and the Department of Pathology and Cell Biology at Columbia University Medical Center. Dr. Yiping Han was my advisor, board of undergraduate advisor for my independent research class. She's also really wonderful. She's a professor of microbial sciences and dental medicine and microbiology and immunology at also Columbia University Medical Center. And Dr. Brent Stockwell, he's also really fantastic. He was my biochemistry professor, and he is a professor in the departments of biological sciences and chemistry. He's a director of the Chemical Probe Synthesis Facility. He's the co-director of High Throughput Screening Columbia Genome Center, and he's an early career scientist in the Howard Hughes Medical Institute. So many thanks to all of my research advisors, and especially Dr. Wang, Dr. Han, and Dr. Stockwell. So now we're going to take a look at the logistical details of biology proposals and some additional tips. So this section of the presentation is largely based on the talk that I went to in 2018, given at Columbia, and the talk was made by Dr. Tanya Dragic, the director of the Grant Advisory Service at the Albert Einstein College of Medicine. So I'm going to go over briefly like an overview of the sources of funding. So there are government sources of funding with the NIH, the National Institute of Health, NSF, the National Science Foundation, and DoD, Department of Defense. The philanthropic sources of funding, where it funds specifically projects, but not like people. And I'll go over sources that fund people in a little bit. But the philanthropic sources of funding that funds projects are Gates Foundation, American Cancer Society, and Susan Komen Foundation. And the sources of funding that fund PIs, principal investigators, or the heads or leaders of research labs are the Borough Wellcome Fund, the Howard Hughes Foundation. So this is just a taste of the different sources of funding, and by no means is a comprehensive list. So I'm gonna talk about the technical components, sorry, not technical components, the logistical components of these proposals, specifically in reference to NIH grants. These are extremely common in the field of biology. So they have a lot of different grants.
Hey, Jenny.
Yes.
Could you restart this slide? There was a little bit of static, and I don't think they're going to be able to hear what you just said.
Okay, got it. Sure. So I'm going to be mostly concentrating on NIH grants for this section of the presentation. They're extremely common in the field of biology. And we're going to take a look at that as an example of the logistical details of biology proposals. And grants are basically roughly the same thing as proposals. So the writing a grant is like essentially writing a proposal to propose a particular project, and it usually involves like research funding. So with this project, you would get the funding, and the funding would be called grant money. So that's what grants are. So the research grants are the R series, and two in particular I wanted to highlight are the R01 research grants, which fund projects up to 5 years, and the R21 research grants that fund projects up to 2 years. So there are also career and development awards, and this is for clinicians or scientists. The research training and fellowship series, which is mostly for postdocs, postdoctoral fellows, and graduate students. And there are the program project center grants, and those are largely for like the major collaborations that happened. And there are the resource grants. These grants are for equipment. So we're going to now focus on specifically the NIH R01 grants, a really big grant in biology. So about 50% of the grants that are submitted are actually discussed like at the round table when all of the reviewers get together. And so the reviewers go through the grants, and each person goes through a certain amount of grants, and then they bring up 50% of these grants to the table for discussion. And about 10% of the grants are actually funded. So this is actually a pretty low percentage, and it's good to know this early on. So one of the major factors that they discuss in deciding who to give the grant money to is overall impact. And the overall impact depends on 5 factors: the significance, the investigator, the innovation, approach, and environment. So I starred the ones that Dr. Dragic mentioned were particularly important for the success of a grant. The significance is what we talked about earlier. The investigator is talking about the history of the person who's proposing the grant. So do they have a strong publication record? Have they made great advances in a similar area previously? Or if they're like more early career, have they demonstrated the ability to really work on a project more independently, etc. So this is investigating the qualifications of the person leading the proposed research project. Innovation we talked about previously. Approach is basically the research methods that we talked about previously. And the environment is about the resources that's available to the investigators. Do they have the proper equipment? Do they have the proper team, or do they have a good team to help them make this project a success? So how to write good grants. This is advice from Dr. Dradrick. And I have starred the ones that she mentioned were especially important. So first, you must be familiar with the scientific literature, in that you've got to know what people have done in the past to be able to set up a good argument for what's a good direction in the future. I mean, that's like kind of intuitive, in that you have to know what we already know in order to figure out what's the best thing to investigate right now. So another good tip is talking to mentors and colleagues about your ideas. So this helps a lot in refining the risk assessment of your ideas, how to make your project less risky and more probable for success based on their experience, what has been more difficult to do. Sometimes like one technique is, although it's established, sometimes it's not very efficient. So it might be difficult to do what you want to do with it if it's not a particularly efficient strategy, etc. Also, make sure to examine trends and funding initiatives. Basically, what are the funding reviewers looking for? What is like particularly popular in terms of like research projects that are funded? What projects do people really want to see right now? So it's important to present rigorous preliminary data. So rigorous preliminary data in terms as in you really want data that can be, that is particularly strong in showing, based on this data, I think my hypothesis is true. And you want to have a good foundation for that to lower the risk of your project and show that there's a pretty good chance that you can do what you're what you want to set out to do. So you also need to create a coherent and convincing story. So you really want to create a story where it will make sense. I mean, you're telling a story of the type of work that you want to do, and it should be something that people can understand easily and also is convincing. By the time they finish reading your proposal, they should be convinced, yeah, this is like a really good idea. Also think about your audience. So you can kind of predict which study session will review your grant. And so thinking about what their expertise is in, and also what they would like to see in these research proposals, is extremely helpful in writing a good grant. So this is a flow chart that Dr. Dradrick had presented that I thought was really, really excellent. So I wanted to present it to you guys. So you start off with the premise, and that's kind of like the current knowledge in the field. And then you go to the problem and the question, like what is the problem? What are the gaps in knowledge, or what is missing from the current knowledge or current innovations, and what specific question do you want to approach? And then you go to your hypothesis, like, okay, this is what I think I would be able, well, not I, just what I think our team would be able to do in terms of discovering new knowledge or creating new innovative products. And then start talking about the significant aims. What exactly specifically do you plan to do in this project to try to test your hypothesis? Either try to create what you want to create or find out what you want to find out. What are the specific goals within this project? And then you go to the research plan. How exactly do you plan to reach your specific aims, to reach the specific goals? And how does this research plan, the conclusions that you reach, the conclusions that you're able to extract after doing this research, how does it add to the current knowledge in the field? And throughout all of this, you want to mention like the significance and the innovation, why this is such a good idea, why it's significant, why it's novel. So some additional tips are give background information where it is needed as kind of like a reminder. So if you mention something before, like a piece of background information before, when it becomes relevant later in like research plans, it's really useful to put in a small reminder like, oh, just reminding you this is the background information that was the impetus for doing this particular experiment. And the preliminary results are meant to show that you can probably do what you propose to do, or that your hypothesis is probably correct. And these results are kind of just bolstering that the probability that your project will be successful. So it's really useful to remind the reviewers of the background significance and innovation throughout the proposal. Like, this is why we're doing it, this is why it's important, and here's how we want to approach it. So also clear and concise language to make sure that your proposal is easily understood. So now I'm going to go into an example of a call for proposals and then take a look at the different projects that were funded as part of this call for proposals. So the proposal that I chose is from DARPA, which I hadn't mentioned in the past, but the DARPA is the Defense Advanced Research Projects Agency, and their mission is to create pivotal investments in breakthrough technologies for national security. So the specific program, the proposal program that I'll be talking about is the PREPARE program. And this PREPARE program, it stands for the Preemptive Expression of Protective, Allele, and Response Elements. So basically their mission is to explore ways to better protect against biological, chemical, or radiological threats by temporarily and reversibly tuning gene expression to bolster the body's defenses against or directly neutralize a given threat. So the premise of this program is that it builds from the understanding that the human body has innate defenses against many different types of health threats, but the body doesn't always activate these defenses quickly or robustly enough to block the worst damage. So to augment the existing physiological responses, PREPARE technologies would provide a programmable capability to up or down regulate gene expression on demand. So that means you would increase or decrease the gene expression as you wish, as you program your technology to do. And this would provide timely, scalable defenses that are proportional to anticipated threats. So this is basically allowing the body to respond faster to things that might damage the body. So service members and first responders could administer these interventions prior to threat exposure or therapeutically after exposure to mitigate the risk of harm or death. So overall objective is to save lives in order to bolster the body's natural defenses against disease. And this program approaches 4 key health challenges. So this is the immediate program goals. It approaches 4 key health challenges: the influenza viral infection, opioid overdose, organophosphate poisoning, and exposure to gamma radiation. So this program really aims for temporary and reversible effects. And this is really key because it's not like a permanent change that you sometimes see in some gene therapies where you change the DNA of the cell and it's like forever changed. So in this program, they aim to preserve the genetic code, so preserve an individual's DNA exactly as it is, and only temporarily modulate gene activity via the epigenome and the transcriptome. So you can think of these broadly as chemical messengers that tell the cell what to do. But the key here is to temporarily modulate gene activity. This is not a permanent change. The epigenome, to explain it a little further, is the changes to the genome in terms of gene expression, so regulation of which genes are activated, which genes are inhibited, and the transcriptome is serves a similar function, but focuses on RNA, so particularly messenger RNA, which transcripts, which messenger RNAs get created, which ones get inhibited, et cetera, et cetera. So the reason why they're looking for this type of temporary and reversible effects is that specific robust protection against many threats at once with an effect that carries less risk. They want to have a specific robust protection against many threats at once. And they want an effect that carries less risk than something that changes the DNA permanently. And it's limited, but it's still tunable. You can still fine tune this in the duration of the effects of these technologies, but you still want it to be entirely reversible. So this is something that would be less risky than a permanent change, but still would allow the changes temporarily to fight the disease. So the immediate program goal is to develop the defenses against these 4 areas, and the ultimate objective of this program is to develop a modular threat agnostic platform solution with common components and manufacturing architecture that can be readily adapted to diverse and emerging threats. So this is a very powerful ultimate objective, and the PREPARE program takes the first step in going in this direction. So the immediate program would primarily use computer cell culture, organoid, and animal models to establish proof of concept. So proof of concept is establishing that the mechanism that you're trying to develop will work. So this is a step before refining this mechanism to really employ it in humans. And the teams in this PREPARE program would work closely with the FDA, the Food and Drug Administration, to make sure that this would be safe for humans when it is finally established and used. And also the teams would work with bioethicists to make sure that everything we're doing is ethical. And part of the bioethic component is in fact the temporary nature of the changes that the PREPARE programs is trying to develop. If you do a permanent change, it leads to a lot more complicated bioethical issues compared with a temporary change that's being used to mitigate the development of disease. So let's take a look at the funded projects in 2019 for this program. So the first is for influenza defense. And so what this program aims to do is to create programmable gene modulators to boost the human body's natural defenses against influenza and also weaken the virus's ability to cause harm by directly neutralizing the viral genomes. So if successful, these teams' approaches would potentially protect against virtually all influenza strains. So influenza strains that either occurred in the past or the future. All of these strains would it would be destroyed by the approaches. In that the humans wouldn't have to worry about influenza ever. It would never evolve to the point where it would be able to attack humans again. So this is very different from the current vaccine, in which you have to get a vaccine every year because it evolves and each vaccine only protects you against a limited sample of influenza strains. This would also, if successful, these projects would also provide near instantaneous immunity in contrast to traditional vaccines. So for traditional vaccines, typically it takes a few weeks for the effect of the vaccine to really take place, for the person to develop the immunity to the disease that the vaccine is for. But in this case, their approaches would instantly immunize the person. So you wouldn't have that lag time. And let's take a look at the 3 projects that were funded for this section, the influenza defense. And so first is a team led by Dr. Robert Debs. So this is in DNAR X LLC, and this team aims to develop a new DNA encoded gene therapy that helps patients fight influenza by boosting the natural immune response and other protective functions of their nasal passages and lungs. So the second team is led by Dr. Phil Santaleglio from the Georgia Institute of Technology. And this team aims to develop novel gene therapies to enable protection against a wide range of influenza strains by delivering mRNA-encoded programmable gene modulators. So this would be things that would upregulate, downregulate gene expression or increase, decrease gene expression by using mRNA-encoded components, and also programmable antivirals to the lungs. This is used for host defense, to boost host defenses and immediately halt viral replication. So the team will also pursue strategies to improve the immune responsiveness and efficacy of current influenza vaccines. And the third team is led by Dr. Robert Finberg from the University of Massachusetts Medical School. And it aims to identify novel host and viral target sequences, including long non-coding RNAs, a very special category of RNAs, which can be used to boost host resilience against influenza. So this project will involve performing innovative CRISPR-based screens on cultured human cells to identify critical host and viral factors using samples from human research subjects and implementing a lung airway model. The second portion of funded projects for the PREPARE program in 2019 is the radiation defense section. So this aims to protect the body from the effects of ionizing gamma radiation. Radiation poisoning primarily affects the stem cells in the blood and the gut. But existing treatments only help to regenerate the blood cells and have limited effect. So it doesn't really actually help stem cells specifically. So there is no possibility currently for prophylactic administration. And prophylactic administration means the administration as a preventative method. So no therapies exist as a preventative method. And most drugs must be delivered immediately following radiation exposure to provide any benefit. So you can't really have that lag time. If you don't get medical help in time, the drugs that are available might not help at all. And also there are no existing medical countermeasures for radiation damage to the gut. So what the teams are working on for the radiation defense part of the PREPARE program is programmable gene modulators that can protect the blood or gut with the possibility of prophylactic administration or interventions. So the two teams that are funded here are by Dr. Harris Wang. Yay! He's my he is my PI, the principal investigator who I work for right now. And so this team aims to develop an orally delivered programmable gene modulator therapeutic. And the multi-modular treatment that the team envisions would take hold in both the gut and the liver, triggering protection and regeneration of intestinal cells, while also inducing liver cells to produce protective cues that trigger the regeneration of blood cells in bone marrow. The second team that was funded is led by Dr. Jonathan Weissman from the University of California, San Francisco. They also aim to develop gene therapies to enhance resilience against ionizing radiation, but this team's approach should result in an intravenous or orally available treatment that activates innate defenses in the gut and blood stem cells for a period of several weeks.
Okay, so now we're going to switch gears a little bit. We've talked a lot about the research process. We've talked about various review papers, or Now we're going to switch gears a little bit. We've talked about various research papers and the techniques for developing a good research process, as well as some of the funding. And now we're going to talk about something else. We're going to talk about review papers. So what is a review paper and how is it different than a research paper? So a research paper is something that we should be very familiar with at this point. We've gone through a lot of them through this semester. So a research paper presents original work. It may reference other works when explaining certain technologies that were used in the development of their idea and methods that they used or assumptions that they made. However, it's primarily a discussion of a researcher's original hypothesis, a detailed summary of what they did and how, and finally, a discussion of their experimental results and what this means for if their experiment was successful or not. This is considered a primary literature source. So this is something that we've talked about a lot so far. Now we're going to talk about review papers. So review papers are very different, but also extremely useful in going from individual research breakthroughs to widely relevant advances in a specific field. So rather than presenting original work, a review paper seeks to synthesize and analyze the many existing technologies in a single area of study. So there's no hypothesis or specific study that is discussed. Rather, many existing papers are referenced and their findings are compared and analyzed. So this is considered a secondary source because it's not presenting original work. And essentially, it provides an overview of a specific topic and looks at many studies that have been previously published. So there are a lot of signs that you can use when you're trying to determine if you're reading a research paper or a review paper. First of all, usually a review paper will have some kind of indication that it is a review. If you look at the citations in it or things like that. Also, a research paper in general has a very specific structure where you start with an abstract that basically summarizes what's going to be talked about, and then we can move into an introduction and methods part where we can talk about the specific research methods that were used for this study, followed by a discussion and analysis. In review papers, rather than having that experimental design setup, there's usually just an introduction that talks about what they're going to be discussing, followed by a brief history of what's happening in the field and a section that discusses the current research and its limitations or challenges. A final thing to look at when you're determining between a research paper and a review paper is that because a review paper is not a primary source and it references a lot of other papers, if you look at the actual references that are cited at the end of the paper, there can be hundreds that are cited rather than in a research paper, which usually has some, but not quite as many. So for context, the research paper that we're going to be going over, the review paper that we're going to be going over today has 90 sources that are cited in it. I've seen up to as many as 250 cited sources, so there can be quite a lot. All right, so the paper that we're going to talk about today is Engineering Bacteria for Cancer Therapy, and it's by Tetsuhiro Hiramoto and Tal Danino. So the topic of interest for this particular review paper is bacterial cancer therapy. So bacteria are naturally present in tumor tissue, and this is because tumors have a reduced immune response in their cores. So some bacteria are able to specifically target tumor tissues for colonization, and this makes them a useful tool for researchers that aim to use these directed bacteria as a way to deliver cancer therapeutics specifically to a tumor site, rather than the widely used systemic delivery form of therapy now. So to do this, we can engineer the bacteria with synthetic biology techniques to give them the properties that make them a successful therapeutic delivery device. So the paper begins by looking at a history of bacterial cancer therapy and its uses before we have synthetic biology techniques come into the picture. So the first instance of bacterial use for combating cancer was applied way back in 1891 when Dr. William Coley injected a patient with live cultures of Streptococcus bacteria and observed remission of inoperable bone and soft tissue sarcomas. So this seems like a very early use of bacterial cancer therapy, but it was not well developed over the next years. Why is this? In general, we believe that it's because it was an extremely risky way of proceeding at that time period. Antibiotics were not widely available, so injecting patients with live bacteria was not a super viable cancer treatment method. As we move forward in time, however, with the development of antibiotics and our better understanding of the ways that bacteria work and our better control over adjusting their aspects with genetic engineering, we can begin to use these in a much safer way. So there has been more exploration in recent times about the effects of different bacteria in cancer treatment, and the paper synthesizes these studies. So some of the referenced ones are the use of Clostridium novi spores for tumor regression in mice and dogs, as well as BCG therapy for bladder cancer. These are just a couple of the ones that were referenced during this history of bacterial cancer therapy portion of the paper, but it gives you a sense of the ways in which the history is synthesized through this paper. So from these studies that we've talked about, they were able to deduce some of the therapeutic effects of bacteria. And there are two main therapeutic effects. So first, bacteria can selectively colonize tumor tissues due to their reduced immune response. This is pretty straightforward. The second effect of bacteria is that accumulation of these bacteria within the tumor combats the cancer cells in that tumor through competing with the cells for resources, secreting toxins, and stimulating an immune response. So especially the stimulation of immune response is important with this because one of the most dangerous things about cancer cells is their ability to go largely undetected and unaffected by the immune system, which allows them to grow unchecked. So after this brief study of the history of bacterial cancer therapy, the paper moves into a discussion of how synthetic bio can be used for improving these mechanisms of therapeutic delivery. So although these studies that we talked about were able to show us a lot about how bacteria is useful in fighting cancer, it's difficult to find a natural bacteria that has the properties that we need for effective cancer treatment. So therefore, we can use synthetic biology to engineer bacteria that have desirable properties. So some of these properties include reduction in strain virulence and endotoxins, which prevent the bacteria that are injected from killing the patient. So synthetic bio, we've obviously discussed, can produce genetic circuits that cause complex dynamic behavior in single cells or in populations. So among these circuits are switches, oscillators, toggles, etc. Think back to the genetic toggle switch that we discussed earlier in the semester. And we can use these SynBio tools to improve the efficacy and safety profiles for cancer therapy. So, for example, we can look at these charts here, which are from a study that shows the effect of bacteria Salmonella typhimurium, which are commonly used for genetic engineering. So these charts show the effect of knocking out a gene called MSBB, which reduces the TNF-alpha induction. So TNF-alpha is an inflammatory cytokine that incites an immune response, leading to apoptosis. So you can see that getting rid of the TNF-A, we can see that in the bottom two charts, which show the survival of mice and Sinclair swine that are injected both with bacteria containing MNSB and bacteria that do not have MSBB minus. So when we look at these charts, we can see that the mice and swine that were injected with the modified bacteria that do not produce TNF-alpha have a much greater survival rate than the ones that were inoculated with the unmodified wild-type bacteria. However, this also has consequences that we don't really want to see. So in the top chart, we can look at tumor retardation by MSBB minus Salmonella. So this has this shows the effects of the wild type Salmonella versus the Salmonella that does not produce TNF-alpha. And we can see that the control has a much greater tumor retardation than the modified ones. So through this modification, we were able to improve the safety of using this bacteria. However, we were not able to see the same efficacy. So there's a lot of more ways that were discussed where we can begin to see the different effects that different modifications have on efficacy and on safety. But improvements are needed to create a model that is both safe and effective. So there is a general principle of modern bacterial cancer therapy. So there's obviously a large advantage of using this bacterial therapy, and that it allows for a reduction of toxicity associated with systemic drug delivery. So when we think about systemic drug delivery, we can think about chemotherapy. And it's in general pretty widely known that the methods that are used to this, while they're effective in killing the tumor, they also produce lots of unpleasant side effects for the patients being treated. So by having this kind of specific targeted delivery, we hope to reduce some of these side effects that patients have to undergo. So a general framework for research in bacterial cancer therapy is described here. So bacteria are in general manipulated by synbio design and introduced by plasmids or other genetic transformation. And then in vitro co-culture systems, which we will discuss in a second, are used to characterize and develop effectively engineered bacteria. To safely deliver therapeutics to the tumor, bacteria are engineered to sense and respond to a tumor microenvironment using synthetic gene circuits. And then these therapeutics are locally released using potentially toxic molecules and immune stimulators. So after we have this good summary of what is happening in the field so far, the general directions that research has been going, the paper discusses then the challenges that we can see that are consistent throughout this field of research. So one large challenge that was investigated is that we're able to engineer bacteria at a higher rate than we are able to test it through throughput animal-based testing. So this slows progress in the research because we have all of these ideas that we can implement to a certain extent, but we can't test them as fast as we would like to. So co-culture of bacteria with mammalian cells is a good platform to study the performance of engineered cells in vitro. However, it has its own limitations as well. So first, what is co-culture? Co-culture is basically growing two different or multiple different types of cells in the same medium. So there are several different ways that we can think about co-culture. If we look at this diagram, we can use it to study natural interactions that happen by placing two types of cells that are normally interacting with each other into a co-culture. You can also engineer synthetic interactions, which we see in part 3, where we have the blue cells are naturally interacting with the purple and the green are naturally interacting with the red. But in co-culture, if we put the blue and the green together, we can see how they will synthetically interact. So there are many different ways to set up a co-culture system, but in general, co-culture allows for the physical compartmentalization of microbes or cells by porous membranes. So any of their secretions can interact with each other and they're in a similar media, but the cells themselves are separated. So this allows us to study the effect that cells have on each other. Some of the limitations of it, though, are that co-culture lacks the spatial orientation and contact between cells that we see in vivo. So the role of 3D interaction is incredibly important for modeling a tumor's response to drugs, because we can observe that 3D in the body and in 3D models, the reaction of tumors to specific drugs is critically different than when we observe its interactions with traditional cell monolayers. So how can we improve this then? There are some available 3D models. Some of them are multicellular spheroids, tumor cylindroids, tumor on a chip, and organoids, which my particular lab uses, that can effectively produce spatial orientation that mimics what you would see in the body, but they in general have low throughput and are only viable for short periods of time. So designing a 3D system that has a high throughput and allows for long-term analysis of bacterial circuit dynamics would greatly improve the testing process and is a solution that is proposed in this review paper. So another challenge that they talk about, which is able to show the nature of what a review paper is able to do by comparing the results of many different existing studies. They take a look next at the conflicting results within existing data in a couple of different places. So one that they reference is what is the role of motility? So there have been conflicting results on the role of motility in tumor colonization by bacteria. So some studies were able to show that overexpression of a flagellum regulator called FLHDC was able to improve bacterial accumulation, while other studies showed that non-motile bacteria were also able to localize within tumors. Obviously, this presents a challenge, and because these studies came up with different results that conflict with each other, this is able to identify a place where we might need to pursue more research in order to come up with a definitive answer in the role of motility. Another thing that they looked at was the role of immune response in bacterial colonization. So one result showed that pro-inflammatory cytokine production is responsible for greater blood flow into a tumor, which enhances the bacterial colonization, while another showed, this is actually the one that is shown below. I apologize, it's a little bit blurry, but that's the way that it was in the paper. This table below shows that reduction in TNF-alpha, which is the cytokine that is pro-inflammatory, did not compromise tumor targeting ability. So we can see this through the ratio of the bacteria in the tumor to liver, which the tumor has a much higher colonization rate. So the paper concludes by saying that bacterial cancer therapy has many advantages over traditional therapies. For instance, the most notable one is the localization of drug delivery and intelligent sensing to a microenvironment and these different conditions of the tumor. And then it also suggests that improvement of efficacy and safety through better characterization platforms and a better understanding of the mechanisms that bacteria use in their different effects could help to accelerate clinical translation of these therapies. So as we can see, this review paper goes through a lot of the existing technologies in order to find places where further research is needed, and also to figure out like what are the most effective methods that have been presented so far. So review papers are a really essential and useful tool for us when we're going from these many individual studies that look at very specific aspects of a field to broadly synthesizing what kind of information they can contribute to furthering the general clinical translation of this field once they are all put together. So a final thing that we're going to talk about today is bioethics. So what is bioethics? So bioethics is a field of study that is concerned with the ethics and philosophical implications of certain biological and medical procedures, technologies, and treatments. We touched on it briefly when we talked about genetic engineering, when we were discussing the use of CRISPR-Cas9. However, we'll go more in detail with it today. So basically, bioethics is the study of ethical, social, and legal issues that arise in biomedicine and biomedical research. Taking bioethics into consideration is incredibly important throughout the entire research process from bench to bedside. It's a particularly challenging field to navigate because healthcare and research affects all people regardless of race, location, religion, or wealth status. So because of this, finding widely accepted ethical solutions to biological problems that satisfy the particular ethical boundaries of each group affected can be a challenge. So first, what is ethics in the broadest sense? Ethics are the moral principles that govern a person's behavior or the conducting of an activity. So not everyone agrees on what ethical behavior is, and there are in general 3 different schools of thought that derive the meaning of ethics. So first of the main groups is deontology. So this was proposed by Immanuel Kant, among others, and deontology seeks to find out what is universally right. It focuses on what duties that you owe to other people and if your actions and intentions are fulfilling these duties. So in deontology, respecting the autonomy of others is a key characteristic. And it seeks to find the definition of what is good by looking at philosophical reason. Second, we have virtue ethics. So in virtue ethics, which is proposed by Aristotle, virtue ethics say that there are certain virtues that we can aspire to and vices that we should avoid. So it's concerned with finding out what kind of person you want to be and has a lot to do with finding out what people can agree upon is reasonable. So in virtue ethics, being reasonable is about the same as being good. Finally, we have consequentialism. So consequentialism has a few different forms. One of them is proposed by David Hume. It focuses on what impact your behavior has upon the world. So is this action causing more harm or more good? In essence, consequentialism aims to do what is useful for society. So in particular, we're going to talk about a subset of consequentialism called utilitarianism. So utilitarianism is a widely used method of ethical reasoning in bioethics. And utilitarianism says that we as a society recognize an action as good if it is useful to us. Therefore, an action that is good is whatever produces the most utility or use. So to illustrate this, we're going to look at a classic ethical debate called the trolley problem. So in the trolley problem, imagine you are the conductor of a trolley, and in front of you there are 5 people on the track that if you do nothing will be run over. However, if you pull a switch, you can change tracks where you will only run over 1 person instead of 5. In the utilitarian view, it would be the right decision to switch tracks. In order to do this, you kill 1 person to save 5. This is the decision that produces the most good. However, we can see this idea applies directly to bioethical debates through the use of animal testing. So in this kind of utilitarian view, some animals suffer so that we can create a cure that saves many. The utilitarian view has a lot of problems, though. By justifying the sacrifice of some to save many, we can justify a lot of terrible things like preemptive war. So this problem also gets trickier when we think about utilitarianism in different settings. So for instance, what if instead of the trolley, you're a doctor and you can save 5 patients, but you have to kill 1 healthy person and use his organs to do it? So this is still choosing to kill 1 person to save 5, yet we probably think about this situation as very different. So this is the utilitarian debate. When we apply this to bioethics, we can think of bioethics as having 4 main principles that are agreed upon, and then their nuances are generally what is debated. So the first one that we have is autonomy. So autonomy is the need to retain the dignity and privacy of participants in clinical studies, as well as the use of volunteered human resources in research. So this mainly serves to ensure that people are informed and consenting to studies and that their human rights are not violated. So we can look at the famous debated case where the bioethical principle of autonomy is relevant when we look at the use of HeLa cells for cancer research. The cells provided breakthroughs in scientific research. However, the original cells were collected from a patient without her knowledge or consent. So it's still okay for us to use them, and does she deserve some kind of compensation? This has been widely debated for a very long time. Next, we have beneficence. So this is straightforward in principle and probably the most direct comparison to the utilitarian debate that we just looked at. So beneficence says that we should weigh the risks and benefits of any action appropriately. And ultimately, if the positive benefits outweigh the potential drawbacks, we can proceed without violating the principle of beneficence. Next, we have non-maleficence, which in essence says do no harm. This is why when we think about the utilitarian debate with regards to organ transplants, like we discussed, rather than the trolley problem, this is why in general we have more of a problem saying that the correct thing to do is to save the 5 patients. This is because when doctors are sworn in, they take the Hippocratic Oath, which says that first they will do no harm. This is why we have a greater problem with this second situation than the first one, and shows more of and is able to demonstrate the nuances in the utilitarian view. Finally, we have justice. So justice is concerned with how can we evenly distribute the benefits of newfound technology throughout society. Everybody is entitled to healthcare, and we want to try as best we can to not make it a competition where people with a higher socioeconomic status are able to reap greater benefits from any newfound technologies. So there are several governing bodies that are responsible for making these bioethical decisions. Starting at the organizational level, the ethical views of your employer or research group play into what technologies they want to explore and feel comfortable using. So then the people providing research funding, which Jenny discussed a little bit earlier, also have a stake in the bioethical decision making and often have guidelines that must be followed for continued funding. So this would be organizations like the NIH. Then the FDA provides broad ethical guidelines for any product that's making its way into the consumer market or clinical trial phases. And there are also, from time to time, appointed bioethical councils in the government, such as the President's Council on Bioethics during the Bush administration. So these are generally made when there are big bioethical decisions that arise and don't usually stay around for a very long time. So there are many good examples of bioethical debates that are probably familiar topics to you all. For instance, we have the debate on the cloning of animals. In general, cloning of animals is a highly contested topic. So on the one hand, it can provide valuable research resources, and the FDA has deemed the meat safe to be eaten. On the other hand, we must consider animal cruelty. So unintended side effects and mutations can arise from cloning. They also make a population more susceptible to disease. So does this cloning make animals a commodity rather than us treating them as a living being? And what rights do animals have within the research process? These are all things that are debated. In general, we try to make sure that animal research is only used when there are no other options, when there is a great benefit to be derived from it, and when we can reduce the suffering of animals as much as possible so that we prevent cruelty. Another debated topic is end-of-life care. So there's debate about the use of drugs for comfort and physician-assisted suicide, but there's also questions about how close to death counts as end-of-life. To what extent do we try to preserve life? What about coma patients that can't be revived but are still technically alive? What do we do in those situations? So there is also a lot of debate about that. However, arguably the most controversial bioethical debate revolves around the use of genetic editing in humans, and no better is this represented than in CRISPR babies. So in 2018, He Jiankui became the first scientist to create the CRISPR-edited human babies, and in 2020, he has been sentenced to serve 3 years in prison for illegal medical practice. So this is a great example of the significance of bioethics in research. So what did he do? He modified a gene called CCR5 in human twin embryos to try to engineer immunity to HIV. There is a naturally occurring mutation in some humans called CCR5 delta 2. This is a deletion which provides immunity from the disease. However, he was unable to successfully reproduce this exact mutation and instead produced a similar mutation in the gene. There is a huge host of problems with this research project. So first, because he was only able to produce a similar deletion and not the exact replica of the CCR5 delta 32 mutation, he created a human genotype that has never been seen before, without testing it first to see if the mutation would actually provide immunity in vitro before implanting the embryos. Theoretically, this mutation will confer resistance. However, we cannot be absolutely certain before we test it out, and we can't be certain then that it will not produce unintended consequences that could be harmful. He used CRISPR editing, which, as we have looked into over the course of the semester, can produce unintended mutation. So while the theory behind his modifications is sound, we can't be absolutely sure that it will produce the intended effect without testing it. So he tried to justify his work by saying that it has the potential to solve the HIV epidemic. So this defense was not incredibly well received as a strong defense of such a radical type of research because it's not a currently viable solution at this point in time. In the future, who knows, we could end up in a position where we see widespread genetic engineering use in humans. However, at this point in time, it's not an incredibly strong defense for what he did today. So Then there is the issue of consent with the doctors, parents, and the embryos. So first, in the research documents, the fertilization doctors and the obstetrician were not referenced as part of contributors for this study. So that has become a large subject of debate where we can try and ask if these doctors really knew what they were participating in when they agreed to help with the fertility of the parents. Then there's the question of whether the parents knew what they were getting into when they agreed to take part in this study. In general, you want the motivations of your participants to be that they are contributing to something. They should know exactly what it is that they're doing, what the risks are. And there has been a lot of debate about whether these parents were just participating because they were able to get fertility treatment from it. And then there's the largest question: what about the embryos themselves? So in general, when we think about the ethical considerations of engineering organisms, there's not a whole lot of thought to the autonomy of the organisms that we engineer. So this is not clearly right or wrong. We should obviously take into consideration what we are doing with the organisms that we manipulate, but as our manipulation usually occurs in bacteria or small cells that we don't really have a huge connection with, we kind of gloss over the autonomy of these beings. However, this becomes a super important part of debate when we're talking about these human embryos. These embryos obviously had no say on whether or not they wanted to be participating in this experiment. However, they will have to live their entire lives with the ramifications of the genetic modifications that they got. So this is a huge topic of debate, whether or not we should take into account the autonomy of these embryos. So the bioethics behind this study are incredibly complicated, but let's consider a different situation. So what if, instead of creating a new genotype, he managed to use CRISPR to create a genotype that was exactly identical to the natural mutation? In this case, a new genotype is not created. Rather, a person's genome is modified to become a more fit existing genotype. Nothing unnatural is being created. However, it's still being modified. In this case, is it still bioethically wrong? Using CRISPR in humans can also present other questions. Does the ability to edit humans lead to eugenics? If embryos can be pre-selected during in vitro fertilization and definitely screened for different kinds of disorders, what kinds of genetic disorders are considered serious enough to terminate life? Would we get gender selection? Do we end up with designer babies? Also, who can afford this technology? This kind of healthcare would be largely available only to the top tier of the socioeconomic status, at least until we have incredible advances in the technology so far. So how is this fair? There is a huge number of questions that we have to address when we're thinking about using CRISPR to edit human beings. As such, bioethics is a very, very important part of that's taken into consideration when designing any type of project that has to do with genetic engineering. So this workshop series is in no way a comprehensive coverage of the entire wide field of synthetic biology. There's much more to learn, but we hope that through these workshops, we've been able to give you some tools and knowledge that can help you in the future if you continue in this research direction. So before we end today with a description of an iGEM project that is a closer representation of what you all might be doing in the near future, I would like to quickly go through and summarize what we have talked about thus far. So in lesson 1, we talked about the history and basic techniques of synthetic biology. So we defined synthetic biology as the application of engineering principles to biology to construct new biological parts, devices, and systems, or to redesign natural systems for useful purposes. We talked about how synthetic biology uses a forward engineering technique with a bottom-up approach generally to design systems for specific function using a combination of natural, modified, and synthetic biomolecules as building blocks. We also then talked about an example as a landmark paper in the field of systems bio when we talked about the Collins toggle switch. I'm sure you all remember this activity. We spent a very long time on it. We talked about how the two sections repress each other and how it confers the ability to toggle between two static states. Then we moved into the topic of genetic engineering. So we briefly discussed the use of sequencing because most of the genetic modifications that we employ today depend on our extensive knowledge of the DNA sequence of the genome of interest. We talked about restriction cloning with the use of plasmids and restriction enzymes as a foundation for genetic manipulation. And then we ended with a look into the mechanisms of CRISPR and gene drive that are essentially molecular scissors that can be passed down to alter a whole population. Then we took our knowledge of genetic engineering for synthetic biology and applied it to real life as we looked at industrial biotechnology. So we discussed the use of creating genetically modified microbial cell factories, which are able to turn different feedstock materials into desired chemicals or precursor molecules. And we also looked at a few applications, including the production of artemisinin in yeast by engineering the yeast with certain plant genes. And then we looked at impossible fuels and textiles. In lesson 4, we briefly diverged from the genetic engineering aspect of the course and instead looked at a different part of synthetic biology, including structural biology, through the lens of biophysics. So we discussed in depth 3 methods of modeling molecules, including NMR spectroscopy, X-ray crystallography, and cryo-EM. So we care about finding the structure of these proteins and complexes because the function and binding of them is strongly correlated with their conformation. And we can see the usefulness of this structural biophysics when we talked about the study of transposons and the structure of the cascade complex when we looked at different technologies in the last lesson. And then for these last 2 lessons, finally, we have looked at the foundations of project design to help show you the information that we have gone over and help you in creating your own projects. We talked about the difference between pure science and engineering. We talked about the basic steps to create a successful project, various pitfalls that you can fall into, and the ways to create a successful project proposal and apply for grant money. Finally, Jenny is going to go ahead and talk about an example project that represents something closer to what you all might be able to do in the near future.
So I'm going to talk about the Columbia iGEM project from the year of 2017. And this is a project that I was involved in when I was a rising sophomore. So this was the summer before my sophomore year, and it extended a little bit into the fall semester of my sophomore year. So I was pretty close to high school age still then, and didn't know too much about genetic engineering. And I was very much beginning my journey as a budding scientist. So I just wanted to give an example of this project and show you that getting involved in these synthetic biology projects really isn't something that's like you have to wait, I don't know, 20 years for. You really can be able to make a pretty good impact in this field, even at a pretty young age as you're developing into scientists. Switch slides.
Oh, sorry. Right.
No worries. So first I'm going to talk a little bit about the iGEM program. The iGEM program stands for the International Genetically Engineered Program. It's a synthetic biology competition and conference, and it's an annual worldwide synthetic biology event aimed primarily at undergraduate university students, but also includes high school students and graduate students. And you can see, check out the iGEM competition and the program at that website. So I was part of the iGEM team in 2017 as part of Columbia. I was part of a 8-membered team with various mentors. We had 5 senior mentors and 4 junior mentors. So this work is all credited towards the incredible mentorship and the great work of my team. I really appreciated this experience, and it was a great introduction for me into synthetic biology. So the premise of the competition is that you create your own synthetic biology project from scratch. So you have to come up with your own idea for what the project is. Then you build and test the product to approach real world problems, and then you present your work and compete at the Jamboree, which is the annual event, which is the culmination of the conference/competition. So the official program for me ran from June to August. We did a little bit of brainstorming beforehand, and we did some work afterwards to prepare for the competition. But this was a relatively short project. So it ran for a couple of months, and this really shows you that how much you can accomplish even as an undergraduate student. Of course, it was with the great support of all my team members and incredible mentors. And so our project was the siRNA-induced oncogene silencing as a therapeutic for cancer-specific gene therapy. I know, a very long title. And I promise you I will explain exactly what all of this means in the coming slides. So the premise of this is that we were interested in cancer. And we see that with the current cancer therapies of radiation and surgery, chemotherapy, they all have many benefits, but they all have many pitfalls in that although they are relatively effective at killing off cancer cells and removing tumors, one of the problems with these therapies is that they're not very specific. I mean, surgery is a little more specific, radiation can be targeted a little more, but but then with surgery, it's difficult to remove the entire tumor. With radiations, the same problem. It's difficult to remove all the cells of the tumor, and with chemotherapy, it has a good chance of removing all the cells, but it's really not very specific to cancer cells. So we were interested in developing a more cancer-specific therapy, and we also wanted to, then that would be to reduce the toxicity of the treatment. So as Miranda talked about in the review paper earlier, bacteria are actually a pretty good mechanism, and they're like a developing mechanism for treating cancer. And so we were looking at bacteria-mediated therapy that was cancer-specific. So why bacteria are useful? Bacteria tend to localize in tumors, and there's the ability of the probiotics, which is the good bacteria in your gut. They can travel to your cancer sites, and from the studies of Dr. Janino's work in 2015, 2016, there really was no harm to healthy tissue in the mouse models that were studied for this. So the reason why the bacteria-mediated therapy are able to congregate around these tumors is because the tumors have hypoxic and immunoprivileged sites. It's primarily the immunoprivileged position of cancer sites that allow the bacteria to congregate and really reproduce, because in around tumors, the immune system naturally wants to kill off cancer cells. So then cancers create an environment where the immune function can't function as well, but because the immune system can't function as well, they can't get rid of bacteria growing there as well either. So then the bacteria are able to localize in the tumors, in the hypoxic environment or the low oxygen environment, and increase their growth. So we were looking at trans-kingdom RNA interference. So this is a bit of a complicated mechanism, but let me explain. So there's two parts of the system. Basically, you're trying to deliver shRNA into the mammalian cells or the human cells. And this shRNA is short hairpin RNA. It's a small piece of RNA that can inhibit the expression of a gene. So basically what happens is that this shRNA gets processed, and after it's processed, it can congregate with another complex called the RISC complex, and it basically pairs with an mRNA. So mRNA, as we know from the central dogma, that's the key to producing proteins, because with mRNA, then you get translation, and then the polypeptide change forms and the protein gets created. So if you get rid of this mRNA, then you can inhibit the expression of the protein. The protein doesn't get produced. So what the shRNA does is that it goes in with this RISC complex, pairs with the mRNA, and causes the mRNA to get cleaved, to get cut in half. And after the mRNA is cut in half, you can't get the whole protein anymore. And so the protein production for that gene is decreased. And one of the special aspects about shRNA is that it's very specific. So the RNA can only pair with an mRNA that it is complementary to. So you have to make sure you have the right amount of the base sequence in order for the mRNA to get cleaved. So that allows for gene-specific inhibition, and that is the essence of RNA interference. Okay, so now we know that what RNA interference means. What does transkingdom mean? Transkingdom is basically the idea of bacteria invading the mammalian cell. Because shRNA is an RNA. RNA is extremely fragile and it can get degraded very quickly. If you ever work with RNA in lab, like if you breathe on it the wrong way, it might degrade. So it's very sensitive. And as a result, you want a pretty good mechanism to deliver the shRNA into the cells that you're trying to attack. And you can do this via bacteria. So what happens is that there's two genes that are involved, and this is the HLY-A gene, which is a listeria-lysine O, and the invasin gene. So the invasin gene allows the bacteria to bind to beta-1 integrin receptors on the cell membrane. And this causes the bacteria to be endocytosed in an endosome. So the mammalian cell basically gulps up the bacteria and puts it in a vesicle, a special vesicle called an endosome. So then after this is in this vesicle, the pH, the acidity, the pH of the endosome goes down, so the acidity goes up, and that causes the bacteria to lyse. So basically it explodes, and all the contents of the bacteria just leak into the endosome. So now you can imagine, you can engineer these bacteria to be constantly producing a lot of shRNA. The shRNA, because it's constantly produced, there's a lot of it in the bacteria. When it lyses in the endosome, then all the shRNA is now in the endosome. But it's still in the endosome though. It hasn't reached the cytoplasm. The shRNA's got to reach the cytoplasm for it to take effect. And that's where listeria-lysine O comes in. And listeria-lysine O produces pores, basically protein pores, in the endosome, so allows the contents of the bacteria to leak into the cytoplasm. So that allows the shRNA to leak into the cytoplasm and have it delivered in the cancer cell. So when this mechanism was designed about 10 years ago, more than 10 years ago now, wow. And when this mechanism was designed, it wasn't very specific as to where it invaded. So you would assume that bacterial colonization doesn't occur at the type of density elsewhere in the body compared to cancer cells. So you get a higher density of bacteria near cancer cells than other parts of the body. But when my team and I read this study, we were thinking, well, why can't we make it more specific? Because it's still like a little bit of non-specific in that even though there's higher density near cancer cells, there can still be some of this stuff like floating around, and that's probably not good. So let's put a regulator on this. So the regulator that we put on this is the quorum sensing. And what quorum sensing essentially is it's the ability of bacteria to sense their own population density and to not activate certain gene expression until the cell population reaches a specific level. So then we can program the cell to not activate this invasion mechanism until the cell density hits a certain spot, then you invade. So then you can get a more cancer-specific therapy using this bacterial-mediated RNA interference therapy. And this is what is shown on this slide. And the LuxI, LuxR, PLux, this is the system that controls the quorum sensing. I'm not going to go into the details. It's a bit complicated. And as you can see, this then controls the invasin, the HLYA, listeria lysin, and we have a GFP there for our model just because it's easier to detect GFP compared to detecting invasin and listeria lysin because you can just see GFP fluorescence. So this is placed in an operon, so then all three of these genes are created one by one. Now we have the invasion mechanism being controlled by the quorum sensing, and now we have the RNA, shRNA on a separate plasmid. And one of the reasons why I wanted to do that is we wanted to create an extremely high production of shRNA, but we didn't want as high a production level of the invasion genes because they are pathogenic genes, so they're more dangerous. But the shRNA, it's fine. So the shRNA, we put it on a high copy plasmid with the puck ORI. This is a high copy number plasmid and high production of shRNA with a T7 promoter enhancer. All of this basically means that we put an engineering mechanism that allowed us to produce a very high level of shRNA. And we used the original design of the shRNA transkingdom mechanism in which they used a restriction sites for cut and paste mechanism. That way, the shRNA that you want to make, you can just switch it out using restriction enzymes. So the specific genes that we were able to target during my iGEM year was EGFR and EGFP. You might wonder what is GFP doing in there? What does it have to do with cancer cells? That was kind of a proof of concept because it's much easier to detect GFP levels than levels of other proteins because of the fluorescence aspect. So that was our proof of concept, and then we applied it to EGFR. And the intention was to use this for more genes, but then because of the short nature of the program, we were only able to get to the EGFR. So one of the things that we wanted to make sure is that producing the shRNA in the bacteria doesn't actually inhibit the growth of the bacteria because we didn't want the production of the shRNA to be toxic to the bacterial cell. So we didn't find theoretically any reason that the shRNA would be toxic to the cell, but as we had mentioned before, you don't know what will happen unless you test it. So we wanted to test it just to make sure that indeed it does not harm the bacterial growth. And you can see here, so the green line and the brown lines are the two different strains of bacteria, one with the shRNA producing mechanism, one without the shRNA producing mechanism, and they overlap quite well. And that means there is no defect in growth as a result of shRNA production, which is good. So you can see that the brown curve is actually slightly lower than the green curve. The green curve is the strain of bacteria without the shRNA production. The brown curve is with, and you can see it's slightly lower. It grows a little bit more slowly, and that's part of the metabolic burden of producing this shRNA. But as you can see, the metabolic burden is pretty minimal. So I don't show another set of results here, but we also tested the quorum sensing circuit to make sure that it worked. We were using the quorum sensing circuit from Dr. Danino's work, and we were able to verify that it works. And now we wanted to see whether our shRNA would work in that does it in fact knock down the expression or inhibit the expression of our target gene. So this is the experiments that we did with the shRNA that targets the EGFP. So we used liposomal transfection induced with induced and in vivo shRNA. So what we did was we had our bacteria produce a ton of this shRNA. We extracted the shRNA, and then we used liposomes to fuse them. Basically, they're small lipid droplets that you can use to deliver shRNAs into mammalian cells. So then we assessed the knockdown effect, the decrease in GFP fluorescence. As a result of this, you can see the PBS is our negative control. So that's just the cells that express GFP. The cells had already been engineered to express GFP. So the mammalian cells expressing GFP with no shRNA. We purchased an siRNA, which is basically a pre-processed version of the shRNA. And this commercial siRNA was designed to specifically target the EGFP gene that was in our mammalian cells. And as you can see, it actually didn't work that well to test the knockdown of the GFP, but our shRNA, which is the third bar, it produced statistically significant difference in the GFP expression, and clearly you can see that there was a decrease in GFP expression, and this shows that our shRNA was effective in knocking down gene expression. So the original objective was to then, now that we know that the quorum sensing works, we were able to produce the shRNA. We were able to show that our shRNAs can successfully inhibit gene expression. The idea was to put all of that together with the invasion mechanism. So that would have been the next step. But because the program was relatively short, we didn't get to that next step. So we had all the working parts. And then the remaining step was to put it all together with the quorum sensing, controlling the invasion, and then the production of shRNA and delivery of that shRNA into our mammalian cells. This is an example of what you can potentially do even as an undergraduate student. This was a really great introduction to synthetic biology. And the reason why I present this is to show that the possibility of getting involved in these synthetic biology projects are really not far off. I mean, we were a team of mostly rising sophomores with one rising senior. So we were a pretty young team, and we still were able to do this in a relatively short amount of time, which is a couple of months. So I really would encourage you, if you're interested in these science projects, to get involved, because absolutely you can be a big factor in helping develop these projects and make these projects a success.
All right. So this concludes the 2020 Synthetic Biology High School Workshop Series. I want to thank you all for following along with us through this uncertain time. We know that this semester hasn't been ideal, as we normally like to teach live lectures, but we hope that you've been able to gain something from this experience, some more knowledge that could be useful to you. And also we hope that you found it entertaining. We hope to be going back to live lessons in the fall, but in the event that we are unable to do it, today we will be sending out a survey to see what you liked and what we can improve upon in our video format. But thank you to everyone who has been participating and coming to these lessons. Also, a big thank you to Jenny, who this is her last lesson before she graduates. You've been a huge help for this entire project. Thank you.
Thank you. You've been a wonderful new project lead.